So I'm Philipp Vermeulen from the Walloon Agricultural Research Center, and I'm in charge of different projects dealing with the plant phenotyping. And so I will show you this afternoon some studies we did regarding plant health, and I will show you some sensors and methodology applicable for biotic stresses in wheat. And it's work we did together with GEVES in France and with Agroscope in Switzerland. Oops. Sorry. Yeah. So what I will try to do this afternoon is to give you some ways to answer to the following question: how to improve the crop resilience to biotic stress. And so it's what we are doing in the PHENET project by developing alternative methods to assess different stress on plants. And we started this study in the previous project, in the EnviroTyping project, and now we continue this this work in the PHENET project. So I will show you different things, different approaches to assess the wheat disease, in particular the Fusarium head blight on wheat. Using different devices, the handheld fluorometer in fields, the RGB imaging, the multispectral imaging, the hyperspectral imaging, and finally an NIR spectrometer and sorting system for the grain. So Fusarium head blight, what is it and why this choice? In fact, we chose this disease on this crop because wheat is a main grain cereal crop cultivated in Europe. And Fusarium head blight is also an important fungal disease. This fungal disease infects, in fact, the ears at the flowering stage. And you can see here on the picture the symptoms of Fusarium head blight. So you have a part of the ear white and a healthy part in green. The problem is that this disease damages the kernel. So you can see here the healthy grain and damaged kernels. And the main important thing is that this Fusarium head blight produces a mycotoxin, so it's a risk for human and animal health. And yes, we showed this study because fungicide treatment is only preventive and not fully effective for the moment. And breeders and farmers need disease-tolerant varieties. And another topic is also that because the symptoms are observable at the canopy level, and so when we work with imaging, it's easier to take image of something we can see. So for that, we work with different varietal trials, and we inoculate them using spore of Fusarium head blight. And so we have different varieties under inoculated and not inoculated conditions. The classical method to assess this disease is a visual observation at the ear level using the scale here on the right. So you can see that when we have just one spikelet infected on the ear, we assess 7% of severity of Fusarium and so on. The other observation we can have also is at plot level. So in this case, we count, in fact, the number of the ears infected by plot. And so what I will show you now is alternative methods to these classical methods. And the first approach concerns the fluorescence measurements using such a handheld device. So this device is based on the chlorophyll fluorescence. You can make the measure at different growing stage when the ear are yet green, but also at maturity. And so in the EnviroTyping project, more or less 10,000 fluorescence measurements were taken by microscope in the field. The advantage of this method is that this device is low cost, but the problem is that, as you can see on the picture, it's a contact sensing with point-to-point measurement, so you don't have the view on full ear, and it's also time-consuming. And so using such a device, in fact, you acquire value of quantum yield. And here on the graph, you can see that for different varieties, this quantum yield value is different depending on the control in red or the inoculated plants. And so this result can be obtained using trials with inoculation and strong infection. This work, in fact, is also a part of the thesis of Simon Treyer in Agroscope. And so for the next step using this method in the PHENET project, the idea is to assess this application under natural infection in a variety testing network in order to improve the comparability among campaign sites and operators. The second approach is RGB imaging, and it is, I will say, the most promising device for detection of Fusarium head blight. So we acquired in the EnviroTyping project more or less 3,000 RGB images in different countries, in Switzerland, in France, in Belgium, in different trials, inoculated, untreated fungicide or treated fungicide trials, and at different growing stage. And so this technique is based, of course, on the color. As I said before, the healthy part stay green on the ear, and the infected part is white. And the advantage of this method is that it's a low-cost device. You can use your own smartphone to take the picture, and the acquisition is quite quick also. And the advantage is also that in this case, using image, you have a view on the full ear, so you are able to detect early when just one spikelet of the ears is infected. And so what we are doing in the PHENET project is based on this library of RGB images, is to annotate those images using two classes, healthy and infected by Fusarium. JVS developed also different methods, semi-automatic model and automatic model to make the annotation of those images. And up to now, as 800 images were annotated like this. The next step is to develop prediction models. So different models have been developed, and one of them has been selected. And so you can see that using this model, we have a performance higher than 80% of white classification. And the work was done using detection of spike as a first step, and on the other side without the detection of spike. But the correlation between the visual scoring and the prediction is more or less the same. So then when we have a model, the idea is to apply this model on the RGB image we acquired in the field, in the plots, in order to assess the resistance of the variety to Fusarium. And so using the model, you can have a mask showing just the ears on the image, and another mask showing you the Fusarium pixel detected, and by just a ratio you have the percentage of Fusarium on your ears. And so this model was applied on different image issue from different smartphone, camera, and so on. And with the same model, we were able to have the same results with the same level of correlation. And so then to facilitate the extraction of the results, a small user interface has been developed by GVS. So you are able to see for each image the predicted image with the ears infected by Fusarium, and you have directly, automatically, the results with the percentage of Fusarium and blight. And if you take 5 or 10 images on your plot, you can also analyze all the images together in order to have in extraction on an Excel file the full results you can after that compute and analyze. Another point regarding those RGB library will be to contribute to the Global Wheat Dataset Consortium. So it's a consortium aiming to improve the precision phenotyping by assembling different data set of images. And up to now, there is already a data set for head detection, another data set for full semantic segmentation, so for the segmentation of the different organs on the plant. And what we will provide with the library of RGB Fusarium and blight images, it's a new module with such images in order to develop new models for the detection of Fusarium. And so the next step in the PHENET project will be to consolidate the guidelines document for image annotating for assess biotic stress. That will be also used for the annotated image. And then the idea is to update and to develop the model and use those models to extend the portfolio of agronomic traits on existing phenotyping device, such as the mobile-based rapid phenotyping application developed by Wageningen. And so it was an application developed in the framework of the INVITE project for traits on tomato. And the idea is to adapt this application for other species and for other traits. And one of them will be the Fusarium and blight on wheat. Another tool is also, as David talked about, the literal stick developed by IFEN. So the idea is also to develop model using this device for the detection of Fusarium and blight. The third approach concerns the visible NIR multispectral imaging. So it's something developed by JVS in France using a multispectral camera with 4 wavelengths, 3 in the RGB and 1 in the near-infrared wavelength range. And so you can see here on the right different setup showing you a setup in the lab to take images on different ears, and the same camera used in the field with a different vector. And so during the INVITE project, a lot of images were required in laboratory and in field. This method is based mainly on the chlorophyll content, because we are in the visible range. And we can use this technique when the ears are green and until 550 degrees the post inoculation. The advantage here is we're working with images in proximal sensing. We are able to have a specific detection for Fusarium against microdocium or yellow rust. You are able also to have early detection when just one spikelet is infected. But the problem with this method is the high cost of the device and the time consuming for the acquisition. So here you have some results in laboratory showing you the different ears with different level of infection. And from that, we develop models in order to detect by pixel, the pixel detected as healthy or infected. And then we can have such results as showing in the graph, the relation between the severity, the visual scoring, and the predicted value. You can see that here we have quite a lot of false positive, but it was a first result we obtained. So the same technique was used also in field, in frontal view. So from the images, what GBS did is to make some annotation on the first row of ears and on Fusarium infection. And then based on that, the two algorithms were developed, one for ear segmentation and one for Fusarium quantification. And then here on the right, you can see the results on one side showing the severity assessed on the RGB image and the predicted value. And so in this case, in fact, the results are good enough to make the assessment of Fusarium classification for the different susceptibility to Fusarium and blight. And the next step here in the PHENET project will be to develop some semi-automatic annotation method in order to assess the biotic stress. The fourth approach concerned hyperspectral imaging. So in this case, we obtain absorbance for 100 wavelength range instead to 4. So we can have also different imaging system working in the visible or in the near-infrared. In our case, we use the near-infrared camera for the Fusarium detection on ears, and you have here the setup for the laboratory with the plate and the wheat ears moving under the camera for the acquisition. It's a line scan camera. And the same camera was used on the grand tree with an translation axis in order to take the image on the canopy of the plots. So in this case, using an IR, but the method is based on the water content. And we can use this technique during the full growing stage of the plants. In our case, with this setup, we can analyze more or less 1 square meter by plot. So you have an idea of the infection on quite a large area of your plot. But the problem is the same as the multispectral imaging, is the high cost and the time consuming for that. Here you have some results in the lab using multivariate model PLSDA, and we were able to have a good performance of the model with sensitivity and specificity higher than 90%. And you can see here on the graph that we were able to differentiate, in fact, 4 classes of severity from healthy to full infected. Here you have the results obtained in the field. So in the field, we have an image taken at the top of the canopy. And here on the predicted image, you can see the healthy ears and the ears infected by Fusarium. In our case, during this trial year, we had, in fact, not only Fusarium, but also Tecal disease. Tecal disease is another disease giving the similar symptom to Fusarium. And so in this case, we were not able to distinguish 2 diseases, but we had good results regarding the overall stress. All this work was also a part of the thesis of Damien Vink. And so in the PHENET project, what we intend to do is to transfer the vector from the gantry to a tractor platform with some adaptation in order to control the natural light. So we will build a blackout box for that. And we will also adapt the angle of the acquisition in order to have a better view of the length of the ear. And other things is also to improve the specificity of the models, but also to have models we can apply directly in the field in order to have a prediction in real time. And so that will be done also with an IT company in Belgium called CETIC. The last approach is not on the plants, but on the grain after harvesting. And so the idea here is to assess the damage on the grains due to the Fusarium. And so we have in our institution a Q-sorter device able to analyze kernel by kernel. And so you can see here the system to feed the kernel on the conveyor belt. You have here an RGB camera, spectrometer, and based on the data and the model we can develop, you are able to sort the grains according to different classes. And so, for example, here you have different topics. You can make a sorting based on the morphological data, but also based on the biochemical characteristic as the protein content, for example. So at this stage, you can see here the technology readiness levels of those different systems. And what we will try to do is to improve this TRL level in the PHENET project. And so another topic also we will do in the PHENET project is to extend, in fact, the use of those technology for other diseases on the wheat, but also other diseases on the leaves. And so that will be done using the perch phenomenon, for example, in GWS, but also the imaging systems. And so just I would like to thank all the people working in different projects and different institutions. Thank you.